Hello, my name is Kiara de la Cruz, and I am going to be talking about the molecular and cellular mechanisms of tooth development, homeostasis, and repair. So a little bit about the introduction. During evolution, tooth morphology tends to increase in complexity while tooth number decreases. So the upper portion of the mammalian tooth is known as the dental crown, and the portion buried inside the jawbone is called the dental root. There are four main types of mammalian teeth. Brachydont, commonly seen in omnivores, have a low crown to root ratio with no crown extension into the jawbones after eruption. Mesodont and hypsodont is when a portion of the crown is reserved within the jawbones. And hypsilodont, which are ever-growing teeth, are highly specialized and erupt continuously throughout the animal's life, with a large percentage of their crowns remaining buried inside the jawbones. These types of teeth are what make up the incisors of rodent lineages, and they preserve their dental stem cells and are able to produce various cell lineages throughout the adulthood to support the continuous formation of the crown. So the objective of this experiment was the tooth provides a suburb system to study the molecular mechanisms and cellular functions that govern organogenesis during development and homeostasis. So in the study, they focused on how the regenerative capability of hypsilodont teeth is maintained by dental stem cells during homeostasis and tissue repair, and this can improve our knowledge of stem cell-driven organ renewal and facilitate the development of therapeutic treatments for patients with dental diseases. So some background information. This is the schematic of the mouse incisor development. So the development of the mouse incisor initiates at around E11, where the dental epithelium, where the green is, invaginates into the mesenchyme, which is the orange, forming the dental placode. Let me zoom in. Placode at around E11.5. A population of non-mitotic cells within the placode accumulates to form an early signaling center termed the initiation knot, which guides the formation of the tooth bud. A vestibular lamina forms adjacent to the developing tooth bud during the bud to cap transition, and the signaling activity of the IK switches to the enamel knot. At the cap stage at around E14.5, lingual and labial cervical loops, which is the LICL and LACL, form on each side of the EK in an asymmetrical pattern along the labial-lingual axis around the dental papilla. And the continuous folding further divides dental epithelium into inner, which is IEE, and outer OEE enamel epithelium, which is surrounding the stellate reticulum region. And the development of the LACL proceeds throughout the Bell stage, whereas the formation of the LICL is disproportionately slower on the medial side, and Hertwig's epithelial root sheath, which is also known as HERS, then forms at the lingual side or postnatal day. So this is just showing how the pulp is formed and the different root analog of it and the crown analog. Okay, so some of the methods include EDA, which is it is a type of ligand that controls the site of the primary EK, which forms at the basal layer of the epithelium, sending signals to shape the tooth bud into the cap stage 2. Mice with mutations in EDA have small EKs exhibiting defects in epithelial invagination and CL formation, which leads to flattened cusps. So a group of SOX2 expressing epithelial cells residing in the CLs has been identified as dental stem cells, which of course supports the continued growth of the tooth and gives rise to all dental epithelial lineages during the formation of both rooted molars and ever-growing mouse incisors. So the epithelial deletion of SOX2 using either SHH CRE or PIX2 CRE pathways leads to abnormal development of mouse posterior molars or incisors, indicating that SOX2 expression is required for both tooth development, cell proliferation, and differentiation. So a little about the SOX2 stem cells. From figure A through C, the illustration of SOX2 expressing stem cell compartments in monophyodonts. In A, they are compared with diphodonts, B, compared with polyphyodonts, and C, which exhibit different rounds of tooth replacements. So in A, in the developing mouse molar, SOX2, which is in green, is expressed in the cervical loop, and the transient rudimentary successional dental lamina, which is the RSDL. And with the initiation of crown eruption, SOX2 cells are lost along with dental epithelial tissue. So the epithelial activation of WNT signaling, which is the red arrow, results in an ectopic tooth bud formation at the RSDL, which is the black arrow, and lacks SOX2 expression. So by contrast, deletion of SOX2 only affects the formation of the posterior molar, which is M2, without obvious effects on the first molar, M1. In B, the human molar, the case of diphodonts, as the initial set of deciduous teeth grow, the replacement set of permanent teeth forms along the dental lamina. And then with the beginning of the root formation in the deciduous teeth, the dental lamina degrades and the connection between the permanent and the deciduous tooth buds is lost. And that's why we do not have replicating teeth, because we don't have the tissues to form it. So in polyphyodonts, which is reptiles, the dental lamina persists, ensuring several rounds of tooth regeneration at different stages of development through the lifetime of these animals. So there is also another method of going about this. There are dozens of miRNAs. They are differently expressed in the tooth and have been shown to interact with various signaling pathways to govern orthodontic differentiation of primary cultured human dental pulp cells. And the deletion of DICER1, the gene encoding an enzyme required for miRNA processing, leads to a general disruption of tooth formation in mice. And the epithelial deletion of DICER1 with two different CRE drivers results in ectopic formation of mouse incisors. So mesenchymal deletion of DICER1, on the other hand, causes defects of varying severity depending on tooth location and type. So future studies will be required to establish a more precise expression profile of miRNAs during early tooth development, as well as during homeostasis and injury repair in adult dental tissues. But with the advent of single-cell RNA sequencing, information from different cell populations can be collected to understand further the complex regulation of miRNAs and their interactions between miRNAs and signaling pathways. So this is just a different form of how tooth development can progress. Finally, there are results of the dentin repair in the human molar. So the schematic of a human molar showing dentin mineralization around the dental pulp, enamel deposition on the tooth crown, and cementum coverage around the tooth surface. The tooth is attached to the alveolar bone via periodontal ligaments. So the dental pulp houses blood vessels and nerves, and various types of stem cells, such as the dental pulp stem cell, or DPSCs, periodontal ligament stem cells, PDLSCs, and stem cells from the root apical papilla, or also known as SCAPs, can be found in the dental pulp and periodontal ligament around the tooth root. So mild injury induces reactionary dentinogenesis, which stimulates increased dentin secretion by odontoblasts. By contrast, reparative dentinogenesis takes place in response to severe injury and requires the recruitment of dental progenitors, which differentiate into odontoblast-like cells to fuel dentin production. So bioactive molecules released from mineralized dental tissues, as well as growth factors produced by recruited inflammatory cells, and can promote both odontogenesis and tissue repair. So the limitations of this study, it includes not fully knowing the mechanisms regulating dental epithelial mesenchymal interactions and cellular activities during progenitor migration and cell fate decisions. Also require in-depth investigation. So I took a different approach to this experiment because it wasn't something that was completely new. It's just something that needs to be built on based on this information, like for example knowing of the SOX2 genes and the miRNA. So that's why there weren't so many results or different findings for this. It's just a little, I guess you can say, eye-opener for future studies. So overall, the discussion is that Tooth organogenesis is a complex process involving the repeated utilization of several pathways that can trigger different effects in distinct compartments of dental tissues. A further study suggests looking into how cells tell different signals apart and what mechanisms are used to achieve specific lineage commitment between cell populations. And ways in which the study has added on to previous knowledge is through modifying cell culture conditions that can stimulate the lineage commitment of dental pulp stem cells toward osteogenesis or parasite-like cells. So yes, again, I wanted to explain this more on the topic that there are different alternatives to possible tooth repair, even though we do not have the set of teeth that other mammals can constantly grow or keep within their roots. And that's why scientists are working on this. And I chose this topic as well because I wanted to be a dentist and I feel it was really important. Also, I wanted to get a different aspect of the molecular and cellular side of this field. So I hope you enjoyed my presentation and thank you for watching.